Welcome to the Deep Dive. Today we're tackling something of a puzzle in the world of medicine.
A big one.
Right. Even with all these incredible leaps in science and tech and, even how we manage research, it feels like we're getting fewer new drugs for all the effort.
That's really the core of it, isn't it? I mean, we have this amazing arsenal now. Things like combinatorial chemistry, making huge libraries of molecules.
Yeah, and sequencing DNA super fast, getting detailed protein structures.
Exactly. High throughput screening, testing, countless compounds, a whole field of biotech. You name it. But when you look at the actual numbers,
The number of new drugs approved for every $1 billion spent on R&D.
it's been going down, stubbornly down.
And there's that name for it, right? Room's law. Moore's law backward.
Here, yeah.
Moore's law is like exponential growth in computing. Room's law. It's the opposite for drugs. Since 1950, roughly, the number of new drugs per billion R&D dollars. It's halved every 9 years or so.
Let that sink in. Halved every 9 years.
In inflation-adjusted terms, that's like an 80-fold drop. It's kind of staggering, actually. Makes you wonder if we're going backwards sometimes.
Precisely. And our mission today, really, is to dig into why. Why is this happening? We've got some great analysis looking at the reasons for this decline.
Despite all that stuff you mentioned, better understanding of disease, new drug targets, better management.
Right. Despite all that progress.
Okay, let's unpack it.
Yeah.
The first big reason they point to, it's got a great name, the better than the Beatles problem. That's quite an analogy. What's behind it?
Well, think about it like this. Any new drug coming out today isn't just appearing out of nowhere, right?
Right.
It has to compete with everything that's already available. And a lot of that is now generic, cheap.
Okay.
So it's like, imagine you're a new musician today. You're not just competing with other new bands. You're up against, well, the Beatles, the Stones, everything. And most of that history is available practically free on Spotify.
Ah, okay. So people aren't going to switch to the new thing just because it's new.
Not necessarily. Especially if the old thing works well and costs pennies. Why would your doctor or your health system pay a premium?
That makes a lot of sense. So a brand new drug, even if it's good, has to be way better than, say, cheap omeprazole for ulcers or a generic statin.
Exactly. Significantly better. Or maybe safer. Or much easier to take. But the bar is higher. Take anti-ulcer drugs, for instance. Perfect example.
Okay.
There's a newer class. Potassium-competitive acid blockers, 15, 20 years ago. These could have been massive blockbusters. They seem safe, effective.
Yeah.
But today, they have to compete with super cheap generic H2 blockers and proton pump inhibitors. And those generics work really well for most people.
So the market for the new drug just shrinks dramatically.
Right. Health systems will likely only pay for it if the generics fail. That really limits the economic value,
So this better than the Beatles thing, it actually changes where companies decide to do their research.
Absolutely. It pushes them towards diseases that are harder to treat, where there aren't lots of good, cheap options already.
Like cancer or autoimmune diseases.
Exactly. And the data shows this. Pomoli and others found R&D projects shifting away from areas with lots of existing drugs, think antibiotics for simple infections, or hormone therapies, and towards these tougher areas. Areas that historically have had lower success rates in clinical trials.
It's so different from, fashion, where
Yeah.
last year's stuff is just gone.
Or publishing, yeah.
Yep.
People want the new bestseller. But medicine's different. Your doctor isn't going to ditch metformin for diabetes just because something new comes along, unless it offers a really big advantage.
And it's important to stress, this isn't quite the same as the low-hanging fruit idea.
Right. Good distinction. Low-hanging fruit is about the easy targets being gone. Better than the Beatles is about the reduced value of hitting even difficult targets. Because good stuff already exists cheaply. The economic incentive is lower.
Okay, so the bar is higher clinically and economically. That leads pretty naturally to the second point, doesn't it? The cautious regulator problem.
Yeah, they're definitely related. Over time, agencies like the FDA have become, well, more cautious, more risk-averse.
Meaning they demand more proof, more data.
Exactly. They keep raising the standards for approval, which, drives up R&D costs, makes things more complex. It's like a ratchet.
A regulatory ratchet.
Yeah, things tend to get tighter, stricter. It's pretty rare for regulations to be significantly loosened.
Is there an example of a specific test or rule that maybe illustrates this?
Well, the analysis mentions the Ames test. It checks for mutagenicity, potential to cause cancer. Introduced for safety, obviously, but some argue it might be a bit vestigial now, maybe screening out some potentially okay drugs without adding that much safety anymore. It's debatable, of course.
And then there was that huge shift back in the 60s, right? The Kefauver-Harris Amendment after thalidomide.
That's right, a watershed moment. Before that, companies mainly had to show a drug was safe. After 62, they had to prove it worked too. Efficacy became key, and safety hurdles went up.
And you actually see a dip in R&D efficiency right after that.
You do, yeah. Makes sense, doesn't it? New, tougher rules, more work needed.
But then things picked up again in the mid to late 90s. Efficiency went up for a bit.
Yeah, a temporary reversal. Likely two main things driving that. First, the FDA started clearing a backlog of applications after the Prescription Drug User Fee Act (PDUFA) came in.
That's where companies pay fees to fund the review process.
Right, and it sped things up considerably. Second, there was the rapid approval of several HIV drugs, intense patient advocacy, the sheer urgency. It effectively lowered some regulatory hurdles for those specific treatments.
And how does this cautious regulator feed back into the better than the Beatles thing? They must interact.
Oh, absolutely. They reinforce each other. If there are already good, safe treatments available for something,
Like those ulcer drugs.
exactly, then the regulator is going to demand even more convincing evidence that the new drug is not just safe, but significantly better. The bar gets higher precisely because good alternatives exist.
It just sounds like layers upon layers of complexity. You mentioned that Novo Nordisk example earlier. The submission taller than the Empire State Building.
Yeah, it's mind-boggling, isn't it? Just for two new insulin therapies, printed out, taller than the Empire State Building. That gives you a real, physical sense of the sheer volume of data required now.
Okay, so we've got better than the Beatles, we've got the cautious regulator.
Right.
What's the third major factor driving Eroom's law?
The third one is described as the throw money at it tendency.
Okay.
Historically, there was just this clear trend. R&D spending went up and up and up.
Why? Just because it used to pay off?
That was probably part of it, yeah. Good returns in the past. Plus, innovation is inherently chancy, stochastic. Maybe more money felt like hedging bets.
Competition, too, being first to market.
Definitely. And maybe some internal company politics. Bigger budgets meaning more prestige, perhaps. So lots of reasons why spending just kept climbing.
Kind of trying to brute force innovation with cash. Did it work?
Well, Eroom's law suggests not very efficiently. And now, actually, we're seeing a shift. Pressure from investors, from executives. There's a big push towards cost-cutting in R&D.
Because they think they can cut costs without hurting output.
That seems to be the perception, yeah. That there's fat to trim, that costs can come down without the number of new drugs dropping further.
Is that realistic, though? Can you just slash budgets and expect innovation to stay the same?
That's the million-dollar question, isn't it? The worry is, well, if the initial increase in spending wasn't always based on a deep understanding of what drives R&D success.
Then the cuts might not be either.
Exactly. You could end up cutting in the wrong places, maybe damaging the productive parts without really tackling the core inefficiencies. It's tricky.
So throwing money didn't solve it, and just cutting money might not either, if you don't fix the underlying problems. Okay, that brings us to number 4, the basic research brute force bias. This sounds really interesting.
It is. This is about perhaps an over-reliance, an overemphasis on the idea that advances in basic science, especially molecular biology, plus these high-throughput screening technologies, that this combination would sort of automatically guarantee success down the line.
The thinking being, understand the molecules, screen millions of compounds, find the ones that hit the target, and boom, you get drugs.
Pretty much, yeah. A very rational, reductionist view. If we just understand the fundamental bits and pieces and we have the tools to screen efficiently, then clinical success should follow.
Seems logical on the surface.
Yeah.
Where did it go wrong, or maybe where was the assumption flawed?
Well, the data is pretty stark. Despite huge leaps in that early stage stuff, identifying targets, screening faster, better computational filters.
All that preclinical work.
Right. The actual probability of a small molecule drug making it all the way through clinical trials,
Yeah.
that number hasn't really budged much. In 50 years, it's stayed stubbornly low and flat.
While overall R&D costs per drug have soared.
Exactly. So there's a huge disconnect. We got much, much better at the start of the process, but it hasn't translated into more success at the end of the process in patients.
The source material asks a really pointed question here. Have we essentially industrialized the wrong set of activities?
It's a provocative thought, but yeah. Looking back, R&D efficiency seemed higher decades ago, before we had all these genomic targets and massive screening libraries.
What were they doing differently then?
It was, perhaps more empirical, more focus on clinical observation, using animal models to see what drugs actually did in a whole system, not just a test tube, more iterative medicinal chemistry.
Less focus on finding the one perfect magic bullet for one specific target.
Possibly, yeah. The article critiques that single target focus because, well, first, the link between hitting one target and fixing a complex disease in a human isn't always that direct or simple.
Yeah.
Biology has redundancies, workarounds.
Right.
And second, many successful drugs historically turned out not to be super specific. They hit multiple targets. They were more like magic shotguns than magic bullets.
Which might be why those older, maybe less sophisticated seeming animal models were actually quite predictive. They captured that broader effect.
Could be. They give a more holistic view, maybe.
Hmm.
It's interesting. The analysis showed that for first-in-class small molecules approved between 99 and 2008, more were discovered using phenotypic assays looking at the effect on whole cells or organisms than target-based assays.
Even though target-based was becoming the dominant approach.
Right. That's a pretty striking finding. It suggests maybe the older way wasn't so bad after all, in some respects.
And the article contrasts the modern serial filtering approach
Yeah.
with the older iterative chemistry approach. Can you unpack that difference?
Sure. So the modern way often starts with screening a huge static library against one target. You get hits, then you filter those hits sequentially based on various criteria, potency, drug-like properties, etc. Filter, filter, filter. The older way, maybe starting pre-1990s, often involved chemists making and testing fewer compounds initially, but testing them in parallel across multiple assays, looking at potency, but also absorption, metabolism, toxicity, ADME, all at the same time.
So getting a broader picture earlier.
Exactly. And then based on that profile, they'd go back and tweak the molecule, iterate, try to improve the overall balance of properties, not just optimize binding to one target. It allowed for perhaps a more thorough exploration of chemical space.
Like the examples of adrenaline leading to beta blockers or histamine to cimetidine. Those weren't straight lines.
No, they were much more winding, iterative paths based on observing physiological effects and gradually refining the chemistry.
It's also mentioned that biologics, antibodies, gene therapies, they tend to have higher success rates in trials than small molecules. Why might that be?
Several potential reasons. Often, biologics mimic or replace the body's own molecules or target very specific external signaling pathways, so their mechanism might be better understood or more directly linked to the disease effect.
Less guesswork involved.
Perhaps. Also, they've opened up new classes of targets, especially cell surface proteins, that maybe aren't as entangled in the better than the Beatles problem yet. And their interactions might be cleaner, more predictable in some ways than small molecules that get inside cells and can hit multiple things.
So what drove this big shift towards the basic research brute force approach if it wasn't purely about better results?
Well, it was likely a confluence of things. You had innovation seeming to slow in the early 80s. You had the cautious regulator effect growing, the Beatles problem starting to bite, worries about animal model.
So a sense of needing a new approach.
Right. And then you had the rise of molecular biology, this powerful reductionist way of thinking the gene as the key to everything. It was intellectually dominant.
And appealing to managers, automation, high throughput.
Exactly. It looked more systematic, scalable, maybe less dependent on the perceived unpredictability of individual chemists' intuition compared to the older ways, more industrial.
And we have to remember survivor bias, right? We hear about the targeted drugs that work, not all the ones that fail using the same logic.
Absolutely. It makes the process seem more rational in hindsight than it probably is. But, there are positive signs too. Oncology is one area.
Where targeted therapies are working well sometimes.
Yes, particularly where there's a very strong validated link between a specific mutation or target and the cancer's growth. That shows the approach can work brilliantly in the right context. And there's maybe a hint of a return towards more iterative methods and earlier proof of concept trials in some companies now.
Okay, so these four big factors, beetles, regulator, money, brute force, they create ripple effects, right?
Yeah.
The secondary symptoms. First one is the narrow clinical search problem.
Yeah, this follows from the brute force bias. If you're hyper-focused on proving your drug hits target X and causes effect Y, you might miss other interesting, unexpected effects.
Serendipity gets lost.
Potentially. Less broad observation in early human studies compared to, say, the 50s and 60s, when physicians might have noticed unexpected benefits because they weren't just looking for one specific outcome tied to a molecular hypothesis.
Then there's the big clinical trial problem. Trials getting bigger and bigger.
Yes, partly the Beatles effect. Again, if your new drug is only slightly better than the standard, you need huge numbers of patients to prove that difference statistically.
Regulators demanding more data, too.
Definitely. More safety data, data across different populations. Plus, marketing wants data involving lots of investigators makes trials complex. It all adds up to bigger, longer, more expensive trials.
Which connects to the next one, the multiple clinical trial problem.
Right. Because medicine is more complex and because good treatments exist, regulators and payers often want data specific to narrower patient groups or disease stages. You can't just extrapolate as easily.
So you need more trials for narrower indications.
Pretty much. The example given is insulin analogs needing more and more separate, pivotal trials over time to get approved for slightly different uses or populations.
And finally, all this contributes to the long cycle time problem. Just takes longer to develop drugs now.
Much longer. Think about Imberman back in the 50s, relatively quick development. Now, average time is over 9 years. Regulatory caution, complex trials, that front-loading of molecular work, it all stretches the timeline way out.
Wow. It's a pretty sobering picture of the challenges. But the analysis doesn't just diagnose. It offers a potential solution, or at least an idea. The Chief Dead Drug Officer. CDDO.
Yeah, it's a fascinating proposal. The idea is to create a really senior role, like board level, focused entirely on understanding failure.
Why drugs die in development.
Exactly, at every stage.
Yeah.
Their main job would be to produce a really detailed report within, say, 18 months, analyzing the causes of Eroom's law within that specific company.
And share it widely, not just keep it internal.
Right. Share it with the board, shareholders, journals, even regulators. Transparency. And the pay structure suggested would incentivize accurate long-term predictions of R&D productivity, not just short-term hype.
The thinking being, there's loads of pressure for optimism in R&D, right? But maybe not enough systematic learning from the failures, which are the biggest cost driver.
Precisely. The person focused on why things fail might be uniquely positioned to see the systemic problems driving Eroom's law. Their report would try to quantify the decline, rank the factors, assess tractability, maybe even use diagrams to show how the sweet spot for viable drugs is shrinking.
It's a radical idea. So, given all this, what's the prognosis? Is Eroom's law just going to keep going?
Interestingly, the authors say they'd actually be surprised if it continues at the industry level for the next, say, 5 to 7 years.
Really? Why?
A couple of reasons. Some are kind of mechanical. Like maybe the industry is finally tackling that throw money at it tendency, so overall R&D spending might flatten out. If spending flattens, the cost per drug calculation changes, even if absolute output doesn't soar.
Okay, what else?
There's the issue of biosimilars potentially being approved as BLAs through the standard biologics pathway. That could inflate the number of novel approvals without huge R&D costs per approval.
Kind of an accounting effect almost?
A bit, yeah. But the more interesting reason they suggest is a potential improvement in oncology drug development. Better signal to noise.
Because targeted therapies are starting to pay off more predictably.
Potentially, yeah. They point to 2011 having a high number of anti-cancer and orphan drug approvals as maybe an early sign. And orphan drugs. Well, they often sidestep some of these big problems.
Like the Beatles problem, less competition.
Right. And the cautious regulator might be slightly different for rare diseases with no treatments. Trial sizes can be smaller, so orphan drugs are less affected by some key drivers of Eroom's law.
So putting it together, flat spending, maybe some biosimilar inflation, plus a boost from oncology and orphan drugs.
Yeah.
That might technically halt the industry-wide decline in drugs per $1 billion?
That seems to be the argument, yeah. It might level off, but, and this is a huge but, they emphasize.
Does that mean good financial returns for the industry?
Exactly. That's a whole other question. Halting Eroom's law, on paper, doesn't automatically mean R&D suddenly becomes a great investment again. The financial markets are still pretty skeptical, even if executives sound optimistic.
That hypothetical chief dead drug officer would certainly have an interesting perspective on that question.
Wouldn't they just?
Okay, so wrapping up this deep dive, it's really clear that this declining efficiency in pharma R&D is, well, it's incredibly complex, isn't it?
Deeply complex, despite all the amazing science.
Yeah. It's this potent mix of factors.
Yeah.
Proving you're better than cheap generics, proving you're better than cheap generics, the rising regulatory bar, past habits of just spending more, maybe focusing too much on one specific type of basic research approach.
It all intertwines. And
Mhm.
understanding these root causes, I mean, that's got to be step one if we want to figure out how to actually improve things, how to get more effective, affordable medicines to people.
Absolutely. And that really leaves us and you listening with a final thought to chew on. Given these powerful forces pushing against R&D efficiency, what kind of fundamental shifts, maybe in research paradigms, maybe in regulation, what might actually be needed to reverse Ehrum's law, to really build a sustainable innovation pipeline for the future?
No easy answers there.
None at all, but definitely worth thinking about. Thanks for joining us for this deep dive.